It's incredible to see so much interest in understanding where we've come from, where we are now, and the exciting future ahead. We will walk through the history, the breakthroughs, and what gives us hope for patients facing a diagnosis of blood cancer. Hello, everyone. My name is Ran Reshef, and I'm a professor of medicine at Columbia University Irving Medical Center in New York, and a member of the Herbert Irving Comprehensive Cancer Center. I serve as the director of the Cell Therapy program, and I'm part of the Blood and Marrow Transplantation program within the Division of Hematology and Oncology. Let's start with the present and near future. How far have we come, and what am I most excited about right now in 2025 in terms of newer treatments, better understanding of cancer biology, and managing side effects for our blood cancer patients? There is more than one thing to be excited about in today's landscape of blood cancer treatment, but first and foremost, it is the increasing role of immunotherapies. We have gradually moved away from using crude chemotherapy regimens to harnessing the immune system to fight cancer. This means we're seeing treatments that are not only more effective, but often come with fewer or more manageable side effects. The prime example for this is CAR T-cell therapy. It has only been around 15 years since the first CAR T-cells were administered successfully to patients with blood cancers. And today we have 7 FDA-approved CAR T-cells covering a large range of blood cancers and numerous second and third generation CAR T-cell therapies in clinical trials. Other advanced immunotherapies, such as bispecific antibodies, antibody-drug conjugates, and monoclonal antibodies, are also changing outcomes for diseases that were once very difficult to treat, and diseases for which the only therapies once available were aggressive chemotherapy agents. Another thing to be excited about is the increasing precision and personalization of our treatments. We have moved away from one-size-fits-all approaches towards therapies that are tailored to the specific characteristics of a patient's cancer. And we're dramatically increasing the precision of methods that detect residual disease, so we can tell patients with a greater degree of confidence if there is a chance that their cancer will come back after completing therapy. Finally, and importantly, we're getting much better at managing the side effects of all our therapies, improving quality of life during and after treatment, and managing cancer survivorship. One of the best examples for this is bone marrow transplants, which used to be appropriate only for children and young adults because they were challenging to get through safely. But today, the pace of innovation in understanding and mitigating side effects of transplant has made this potent form of therapy much more accessible than before, and we frequently transplant patients well into their 70s and patients with significant underlying medical conditions. Similar improvement in side effect management we're seeing across the board in oncology, and this advancement is truly remarkable. Before radiation and chemotherapy, what was used to treat a blood cancer? It is so gratifying to step back in time and see how dramatically cancer therapy evolved during the 20th century and the beginning of the 21st century. Before the advent of modern therapies, options were extremely limited, often based on the prevailing medical theories of the time, which didn't truly understand cancer at the level of the cells and molecules that play a part in creating it and allowing it to grow uncontrollably. A common way to treat all types of cancer used to be surgery. And while surgery is still today a mainstay therapy for solid tumors, it has fairly limited application in blood cancers, which typically arise in the marrow or in the lymph nodes and are often widespread throughout the blood and immune systems when they're diagnosed and cannot be simply surgically removed. There are specific situations in which surgery does help. For example, removal of the spleen in certain diseases that cause enlargement of the spleen or splenomegaly. While that does not cure the disease, it does lead to significant symptomatic relief and is still used today. Surgeons still have an important role in diagnosing blood cancers because a high-quality biopsy of the tumor is critical for making the diagnosis correctly and remains the beginning of the journey for most patients. Another ancient therapy worth mentioning is bloodletting or leeches. Based on ancient theories of balancing bodily humors or the fluids of the body, which go back to Hippocrates and ancient Greece, physicians would regularly use bloodletting or leeches, thinking they were removing bad blood. Of course, this didn't treat the cancer and could often weaken the patient further. Interestingly, bloodletting has not become completely obsolete. It is still an effective and easy way to remove iron accumulation from the body or red blood cells in cases where the body makes too many of them. For example, in a disease called polycythemia vera. Other remedies in ancient history involved various tonics and substances, but without understanding how cancer is formed and why its growth is poorly controlled by the body, these were usually ineffective. Essentially, before we understood the cellular basis of cancer, there were no truly effective treatments for blood cancers. One important contribution of the old world that stays with us today is a focus on symptom management and supportive measures. In the absence of truly effective therapies, this was the mainstay. Efforts were made to keep patients comfortable, manage fevers, pain, often with substances like opium, not very different from what we do today, and address weakness through rest and various dietary recommendations of the era. While these had no direct impact on the underlying disease, they formed the basis for palliative medicine, which is a critical part of the care for any cancer patient. Then radiation and chemotherapy came to treat cancer. The development of radiation therapy and later chemotherapy truly revolutionized the outlook for patients with blood cancers, which prior to the mid-20th century were almost invariably fatal. Radiation therapy emerged in the early 20th century following the discovery of X-rays by Wilhelm Röntgen, who was a German scientist who discovered accidentally X-rays in 1895. He found these invisible rays could pass through most solid objects, but were blocked by dense materials like bone, allowing him to create an image of his wife's hand, which became the first and very famous X-ray image. Shortly after that, radioactivity was discovered by Marie and Pierre Curie in France. Scientists quickly realized that these new forms of energy could damage or kill cells, and that it would be easier for them to damage cells that are more rapidly dividing, so they could potentially harm cancer cells a bit more than healthy cells. Early pioneers began experimenting with radiation to treat various conditions as early as 1896, and it was found to be effective in shrinking tumors. Lymphoma was in fact one of the cancers where radiation showed very early promise. Physicians in the early 1900s reported that the X-rays could shrink enlarged lymph nodes in patients with Hodgkin's disease, which today we know is a type of lymphoma. By the 1920s and 1930s, radiation therapy had become an established, though still evolving, treatment modality for both lymphomas and many other cancers. This marked a significant step forward, the first non-surgical approach with a real impact on cancer. Understanding proper dosing, targeting, long-term side effects took many decades of research and refinement to reach the sophisticated and precise radiation therapies we have today, a century after the first attempts. The origins of chemotherapy are very different, but similarly fascinating, and linked to a wartime observation. During World War I, sulfur mustard gas was used as a horrible chemical weapon, and scientists observed that it had a profound effect on soldiers' bone marrow, drastically reducing their white blood cell counts by preventing or suppressing healthy hematopoiesis, the term we use to describe blood cell production in the bone marrow. This led researchers to wonder if a related, more controlled compound could be used to target and kill rapidly dividing cancer cells. And this idea remained somewhat dormant until World War II, when the US actually initiated research programs on chemical warfare. At that time, two scientists at Yale University named Goodman and Gilman started experimenting with nitrogen mustard, one of the very first chemotherapy drugs. They were the first ones to administer it in 1942 to a patient with a disease called lymphosarcoma, which is an outdated term for a certain type of lymphoma. And the results were remarkable. The patient's tumors shrank considerably, opening the door to a whole new class of treatments using chemicals to fight cancer. The publication of Goodman and Gilman's work after the war ended ignited intense interest in finding other chemicals that could fight cancer. This was the true beginning of the modern chemotherapy era. Worth mentioning briefly the important work of Sidney Farber, a scientist in Boston who achieved the first remissions in childhood acute lymphoblastic leukemia in 1948 using drugs that block folic acid, a nutrient essential for cell division. This was another landmark achievement demonstrating that cancer cells could be selectively targeted by a chemical. Today, we still widely use methotrexate, which is basically a less toxic version of that first drug that Farber used in the 1940s. The story of chemotherapy's development is extensive, but from these beginnings, researchers went on to discover and develop hundreds of different chemotherapy drugs with different mechanisms of action. For example, alkylating agents like cyclophosphamide and melphalan evolved from those first experiments with nitrogen mustard. Antimetabolites like methotrexate evolved from Farber's work, and many other classes of chemotherapies evolved similarly. An important milestone to point out is the concept of combination chemotherapy, the idea of using multiple drugs together, which may allow synergism, where one drug potentiates the effect of another drug, or as a way to overcome resistance, where a subset of the tumor cells are resistant to a single chemotherapy agent, but a combination may work better to eradicate them. This further revolutionized cancer treatment in the 1960s and beyond, leading for the first time to cures and long-term remissions for many types of cancer, primarily cancers of the blood. Let's talk now about transplants, which represent another major pillar in the treatment of many blood cancers. The concept of a bone marrow or stem cell transplantation is to replace the patient's entire blood and immune system with a healthy one. We use these terms bone marrow transplant and stem cell transplant interchangeably. Obviously, the entire bone marrow cannot be surgically replaced, similar to a transplant of the liver or the heart, but it turns out that the bone marrow, which is the factory that makes our entire blood system and immune system, can be replaced using high-dose chemotherapy, sometimes followed by radiation, to wipe out the blood cancer together with whatever is left from the healthy bone marrow, and then rescuing the patient by infusing healthy stem cells, which can then act as these seed cells to grow an entire new blood and immune system from scratch. Generally speaking, there are two types of transplants. An autologous transplant is where autologous means self. Here we collect the patient's own stem cells, put them in the freezer, and then treat the patient with high-dose chemotherapy, and then give them their own cells back. The goal here is primarily to allow for a higher chemo dose than would normally be tolerated, and this is extremely effective in diseases that are generally sensitive to chemotherapy, such as myeloma and lymphoma. An allogeneic transplant, on the other hand, is a transplant of stem cells that come from a donor, where allogeneic means other. The donor could be someone's sibling or some other type of relative, or an unrelated donor that we can identify in large registries of volunteer donors that contain millions of individuals who volunteer to be on these registries. An allogeneic transplant is more complex than an autologous one because we're introducing an entirely new immune system into someone's body. This new immune system can help fight the remaining cancer. This is often called the graft versus leukemia effect. But it can also potentially attack the patient's own healthy organs, which is called the graft versus host disease, or GVHD, which has the potential of becoming a severe complication of an allogeneic transplant. These transplants are now routinely used for acute leukemias, myelodysplastic syndromes, and some types of lymphoma, and they are known to be a curative approach. The journey of bone marrow and stem cell transplantation is a story of perseverance and groundbreaking scientific discovery spanning several decades. The idea that bone marrow can be transplanted began with animal experiments in the 1940s and the 1950s. Researchers observed that mice exposed to lethal doses of radiation could be saved from dying if they were injected with bone marrow cells from a healthy mouse. This demonstrated that the marrow contained cells that could restore the entire blood cell production. Today, we're very familiar with these cells. We characterize them, we studied them all the time. We call them hematopoietic stem cells. And many don't know this, but the first bone marrow transplant in humans was performed not far from where I am right now at a hospital affiliated with Columbia University, where E. Donald Thomas, who was then a Columbia physician and scientist, performed the first transplant. This was published in the New England Journal of Medicine in 1957. Thomas subsequently moved to the Fred Hutch Cancer Center in Seattle and is certainly considered the founding father of bone marrow transplantation, an achievement for which he was awarded the Nobel Prize in 1990, given that this achievement has revolutionized the treatment of leukemias and many other cancers. Transplants have become significantly safer over the years, and this is due to several factors. Better donor matching techniques. Today we have the capability to conduct HLA typing or tissue typing at a very high resolution and ensure that we have the best matched donor for each and every patient. We have improved supportive care, better antibiotics, antifungals, antivirals to prevent, monitor, and treat infections during that period in which the patient's immune system is still recovering. Better blood product support also helps. It helps the patient cross that period when the new marrow is still not producing enough blood cells. And also better nutritional support and understanding of the specific side effects that may affect the heart, the lungs, the liver, and the kidneys, and are now much more manageable than they used to be. We also have much better strategies to prevent and manage GVHD. I've dedicated much of my career to research graft versus host disease, and this has dramatically evolved over the years, and we now have several therapies for either prevention or treatment of GVHD that made this complication less common and have much less long-term impact on patients' quality of life. Many patients today will come off their immune suppressive medications by 1 year after transplant and won't need to worry about GVHD beyond that time point. Finally, the development of reduced intensity, or what we call mini transplants, which use lower doses of chemotherapy and radiation, made transplants accessible to older or less fit individuals. Now let's move on to speak about targeted therapies. Thankfully, we moved into an era of more intelligent drugs, which we call targeted therapies. Unlike traditional chemotherapy, which often kills any rapidly dividing cell, cancerous or healthy, targeted therapies are designed to attack specific molecules or pathways that are crucial for cancer cells' growth and survival. So they're a lot more specific to cancer than healthy tissue. Targeted therapies go after the cancer-sparing normal cells. Targeted therapies have also become a cornerstone of personalized medicine, where the treatment is tailored to the specific genetic or molecular makeup of a patient's individual cancer. The advantage of targeted therapies is often greater effectiveness against cancers that carry that specific target and potentially fewer of the classic chemo side effects, although they can have their own unique side effects, which requires careful monitoring by a hematologist or an oncologist. Let's look at a few examples. Gleevec or imatinib. This was one of the first game changers. Approved in 2001, Gleevec targets a specific abnormal protein, BCR-ABL, that drives a type of blood cancer called chronic myeloid leukemia or CML. Before Gleevec, CML was often fatal or required an allogeneic bone marrow transplant to treat it. Now, many patients can manage their CML long-term with a daily pill. And while Gleevec is used for some other cancers as well, its impact on CML is the most iconic example of a targeted therapy in blood cancer, and it led to the development of numerous other small molecules that target cancer cells through a specific gene mutation or pathway that fuels their growth. Another example are JAK inhibitors. For patients with myeloproliferative neoplasms like polycythemia vera or myelofibrosis, many have a mutation in a gene called JAK2. We now have drugs that specifically inhibit JAK2, helping to control symptoms, reduce spleen size, which is one of the prominent symptoms of these diseases. The first JAK2 inhibitor was Jakafi, or ruxolitinib, but since then an entire family of drugs have emerged, each with slightly different features, which allows tailoring this therapy better. What about monoclonal antibody therapies? These are engineered antibodies that can recognize and attach specific proteins on the surface of cancer cells. Some mark the cancer cells for destruction by the immune system, while others might block growth signals or deliver toxins directly to the cancer cell. Monoclonal antibodies are targeted therapies, but also mark the transition of oncology toward the development of a new class of drugs that harness the immune system to fight cancer. The last class is antibody-drug conjugates. These are another form of targeted therapy. Antibody-drug conjugates, or ADCs, are like guided missiles, where a potent chemotherapy drug is attached to an antibody that specifically seeks out cancer cells, delivering the payload directly into the tumor while minimizing damage to healthy cells. There have been tremendous advances in the development of ADCs, and work continues to identify new targets for these drugs and developing new payloads that are even more effective while being gentler on the body. Now let's shift gears to another incredibly exciting and powerful approach in treating blood cancers, and that's immunotherapy. At its core, immunotherapy is a type of cancer treatment that uses the patient's own immune system to find and destroy cancer cells. This is one of the most exciting frontiers in medicine in general, not just in oncology. Our immune system is our body's natural defense force, constantly working to identify and eliminate threats like bacteria and viruses. But it is also capable of identifying abnormal or mutated cells, such as cancer cells, and eliminating them too. However, cancer cells can be tricky. They often develop ways to hide from the immune system or even switch it off. Immunotherapy aims to overcome these tricks and re-educate or boost the immune system to recognize and attack cancer cells more effectively. One of the major hopes, and in many cases realities, of immunotherapy is that it can lead to not only better outcomes, but often with different and sometimes fewer or less severe side effects compared to traditional treatments, because it's working more in concert with the body's natural defense systems. Here are some key types of immunotherapy currently used in blood cancers. Monoclonal antibodies we already briefly discussed. Some of them work by flagging cancer cells or blocking their growth signals. Others might be designed to directly stimulate an immune response against the cancer. Checkpoint inhibitors is another family of agents. We know that cancer cells can sometimes put brakes on the immune system to protect themselves. Checkpoint inhibitors release these brakes, allowing immune cells, such as T cells, for example, to attack the cancer more effectively. Checkpoint inhibitors have become a standard therapy, frequently replaced chemotherapy in many solid tumors, and have a more limited but still important role in certain types of blood cancers. Another group of agents are cancer vaccines. While still evolving, the idea of cancer vaccination is to train the immune system to recognize specific targets on cancer cells, much like vaccines do for infectious diseases. Cancer vaccines have been in clinical trials for several decades, and we're finally seeing some signs of success with this approach. We might be getting closer to the first ones getting approved. Now let's move on to talking about CAR-T cell therapy, which is truly a revolutionary approach. I was fortunate enough to be a young faculty member at the University of Pennsylvania when a team of scientists there, led by Carl June and David Porter, treated the first patients with CAR-T cell therapy approximately 15 years ago. I'm also very fortunate to be working today here at Columbia very closely with Michel Sedling, a pioneer in cell and gene therapy who at the same time used a very similar approach in adult patients with acute lymphoblastic leukemia. These initial success stories truly created an entire new field of genetically modified cell therapies. That led to the FDA approval of 7 CAR-T cell products that are successful in inducing long-term remissions and often cures in certain types of blood cancers. How does it work? In broad strokes, we take a certain number of patients' own T cells, a type of immune cell from the blood. We then genetically engineer them in the lab to express what we call a CAR, which stands for a chimeric antigen receptor. It is an artificial protein that specifically recognizes their cancer cells. We can then culture these engineered cells in the lab and create large numbers of them. Over a period of just several days, we can generate millions and billions of these CAR-T cells and infuse them back into the patient. These CAR-T cells then act as a living drug, seeking out and serially killing cancer cells. We also know today that CAR-T cells may remain in the body for many years, and perhaps by doing that, they offer certain long-term protection against cancer recurrence. Another class of agents are bispecific antibodies. These are newer engineered antibodies that have two arms, where one arm grabs onto a cancer cell and the other arm grabs onto an immune cell, such as a T cell. And bringing them together facilitates cancer cell killing. The first bispecific antibody, blinatumomab, was approved for B-cell acute lymphoblastic leukemia, or ALL, in 2014, and more recently, several bispecific antibodies have been approved for use in various types of lymphoma and in myeloma, in many cases replacing old-school chemotherapy strategies. And many more bispecifics are in clinical trials. So you might be wondering, after hearing about all these incredible advancements like targeted therapies and immunotherapies, why has the science improved so much, especially in recent years? It's a great question, and the answer really boils down to a few key but interconnected factors. First one is clinical trials. This is absolutely crucial. Every advance we've discussed was tested and validated through rigorous clinical trials. Patients who participate in trials are true heroes, helping us learn what works, what doesn't work, and how to make treatments better and safer for everyone. Without patients generously volunteering to participate in clinical trials and without the ethical framework that governs them, we simply wouldn't have the new therapies that are now saving lives and improving outcomes today. Each trial builds on the knowledge gained from the previous ones, creating a continuous cycle of improvement. Second factor is, of course, having brilliant researchers. We really stand on the shoulders of countless dedicated scientists and researchers all around the world who have unraveled the complex biology of cancer. It is their curiosity, persistence, and collaboration that drive discovery. And if you are a cancer patient, many of your doctors are what we call physician scientists, people who bridge the gap between the lab and the clinic. They see the challenges patients face firsthand, and they take those insights back to developing new ideas and lead new clinical trials. Of course, we wouldn't be here without technological advancements. Tools like genetic sequencing, gene editing, advanced imaging such as PET scans, sophisticated lab techniques. All of those allow us to understand cancer at a molecular level like never before, and have better tools for the diagnosis, detection, and characterization of cancer in individual patients. We also feel that it's very important to have increased collaboration. Science is rarely a solo endeavor anymore. Scientists, doctors, pharmaceutical companies, organizations like the Leukemia and Lymphoma Society, and many patient advocacy groups are working together much more effectively than ever. Finally, I cannot emphasize enough the role of governments, both the US and around the world, in funding, setting priorities, guiding and organizing cancer research. Furthermore, government plays a key organizational role by supporting research infrastructure, fostering collaboration between institutions, industry, and patient groups, and very importantly, training the next generation of scientists. This foundational support accelerates technological development and ensures that medical research moves forward, ultimately translating into the life-saving therapies we have today. What should we look forward to in the next 5 to 10 years? So I expect the trends we're seeing now would accelerate. We're going to have even more precision. We will get better at identifying the right treatment for the right patient at the right time, based on detailed molecular profiling of each patient's individual cancer. One specific area that is rapidly evolving is the use of circulating tumor DNA, or ctDNA. These are tiny fragments of DNA shed by tumors into the bloodstream. We now have tools sensitive enough to detect and quantify these tiny amounts of tumor DNA. This liquid biopsy approach is helping us monitor how well a treatment is working in real time and detect if the cancer is recurring, sometimes much earlier than traditional imaging, and certainly earlier than waiting for the patient to become symptomatic again. We are already using these techniques in lymphoma, leukemia, and myeloma, and this allows for quicker adjustments to treatment plans. I suspect these methods will become more and more sensitive and perhaps at some point replace standard diagnostic biopsies or imaging studies. We're also moving into the era of even smarter immunotherapies. We will likely see more effective and potentially less toxic versions of CAR-T cells, more bispecific antibodies, and other types of immunotherapies that are emerging and perhaps used earlier in treatment or in new combinations. I would put a lot of emphasis on the idea of combination therapies, which is likely to continue to grow now that we have families of agents, including targeted therapies, immunotherapies, and even old-school low-dose chemotherapy that can work together to attack cancer from multiple angles and prevent treatment resistance. We are seeing this evolve very rapidly in CLL, for example, where chemotherapy that used to be mainstay of therapy has largely been replaced by targeted agents, either alone or in combination. Even more impressively, we are now seeing patients with acute myeloid leukemia, a very aggressive disease, treated with chemotherapy-free regimens due to the excellent efficacy of combinations of targeted agents, something that I did not expect to see in my lifetime. There will be an increase in focus on quality of life and survivorship. We will continue to emphasize how important it is to minimize long-term side effects and ensure patients can live full lives after treatment. We will continue to have improved understanding of the long-term consequences of cancer and its therapies, both on the body and on the mind, and develop better tools to help mitigate some of these consequences. And finally, it is always better to prevent cancer or detect it earlier than to treat it once established. We have a far better understanding of how blood cancers evolve than we ever had. That, together with outstanding advances in diagnostics, such as next-generation DNA sequencing, might allow us to detect some blood cancers or precancerous conditions earlier, potentially allowing for intervention before they become fully established. The ultimate goal is to turn more blood cancers into manageable chronic conditions, or better yet, to achieve cures for as many patients as possible. I would like to thank the Leukemia and Lymphoma Society for producing this webinar, and I wish you all good health.